Scientists today are practically drowning in genetic data. The secrets to so many diseases are hidden right there in our DNA, but actually finding them has been this monumental challenge. So today, we're diving into a new tool that's designed to change all that, a tool that helps untangle that incredibly complex link between our genes and our health.
So let's start with the most fundamental question. How do scientists actually do this? When you have, say, thousands of people with a disease and thousands without, how on earth do you pinpoint the tiny, almost invisible genetic clues that might be responsible? This is seriously one of the biggest puzzles in modern medicine.
To answer that, we basically have to become genetic detectives. The clues we're looking for aren't out in the open. They're buried deep inside our DNA, in the very code that makes us who we are.
Now, these clues have a bit of a mouthful for a name, single nucleotide polymorphisms. But luckily, everyone just calls them SNPs for short. The easiest way to think of them is like tiny one-letter typos in the massive instruction manual that is your genetic code. Most of them are harmless, but every now and then, one can be a really big deal.
And just how many of these typos are we talking about? Well, get this. The average person has between 4 and 5 million of them. Yeah, millions. That's like trying to find one critical clue, not in a haystack, but in a library with millions of books. And here's the crucial part to remember. While the vast majority of these little variations are totally harmless, a select few can actually make us more or less likely to get a certain disease. The entire game is about finding that one important SNP in a sea of millions. But finding that needle in the genetic haystack has, for a long time, been stuck in a major traffic jam, a huge bottleneck. The scientific ideas were there, but honestly, the tools just couldn't keep up with the data. As the paper we're looking at puts it, researchers were constantly fighting an uphill battle, relying on clunky manual methods or trying to juggle a bunch of different software tools. Think about it like trying to build some complex furniture, but you have to get the screws from one store, the wood from a second, and the instructions from a third. It's just a recipe for frustration. And this was the reality. Researchers would have to run their data through one program for a basic quality check. Then they'd have to export that and pull it into a second tool to actually calculate the disease risk. And they still weren't done. They'd then have to use a third program just to make a chart of the results. The end results? Well, the whole thing was painfully slow and super inefficient. And every time you move data between programs, you're just creating a new chance for an error to creep in. Plus, you pretty much had to be a statistics wizard to even make it all work. So, this bottleneck got so bad that a team of researchers finally said, okay, we've had enough. They decided to build a solution from the ground up, one single integrated tool designed to solve this exact problem. They called it GeneRiskCalc. It's a simple name for a tool with a simple, powerful mission to streamline genetic risk analysis once and for all. And its strengths are a direct answer to all those problems we just talked about. It's an all-in-one platform, so no more juggling 3 tools. It has all the important statistical tests built right in. And, maybe best of all, you don't need to be a programmer to use it. It's a website, and it's totally free for anyone to access. So, how did they pull it off? Well, the real magic is in its simplicity. They took that clunky, multi-step process and just streamlined the whole thing. The new workflow is just a dream. A researcher just enters their raw data, then, with a single click, that's it, one click, the tool runs all the essential analyses, and boom, out comes a clear, professional-looking chart showing the results. What used to be a 3-headed monster of a process is now just one simple step. Now, one of the key things this tool calculates is called the odds ratio, or OR. This number is the star of the show. It's a single statistic that tells you how strongly a specific SNP is linked to a disease. If the odds ratio is greater than 1, it suggests that that variant increases the risk. If it's less than 1, it might actually be protective. Okay, so a faster tool is great, but it's totally useless if the results aren't right. So the really big question was, is it accurate? The creators knew they had to prove it, so they put it through some serious, rigorous testing. First, they did a head-to-head comparison. They pitted GeneRiskCalc against those old, established statistical tools. They fed the exact same data into both and checked to see if the new kid on the block got the same answers as the trusted veterans. The result? The numbers were a match, showing an incredibly high degree of consistency.
But you can't just test against another computer program. You've got to use real-world data. So next, they took findings from actual published studies on diseases ranging from migraines to breast cancer. And as you can see, in every single case, GeneRiskCalc was able to replicate and confirm the original study's results. This was the ultimate proof that it was reliable.
So the takeaway from all this testing couldn't be clearer. GeneRiskCalc isn't just faster and easier to use, it's also scientifically accurate and robust. You can trust the results. Okay, so a faster, easier tool is obviously great for scientists. But what does it mean for the rest of us? I mean, why does accelerating this kind of research really matter in the grand scheme of things?
Well, the advantage really boils down to three key things. First, the speed of analysis goes way, way up. Second, it makes the research more accessible to a lot more people. And third, it improves the reproducibility of the science, which makes the results more trustworthy for everyone.
And this is the really big picture. By streamlining the workflow, you're actually democratizing research. More scientists, like clinicians or young researchers just starting their careers, can now jump in and contribute. That means more brains on the problem, which in turn speeds up the discovery of disease-related genes. And that is the absolute foundation for the future of precision medicine. The authors of the paper sum it up perfectly. They say that by integrating all these steps into one single platform, GeneRiskCalc, and I'm quoting here, enhances accessibility, promotes reproducibility, and accelerates research. That really is the ultimate goal, isn't it? Which leaves us with a really fascinating thought to end on. For decades, the pace of genetic discovery wasn't just limited by our big ideas, but by the friction and the clumsiness of our tools. So what happens now that a major source of that friction has been removed? What incredible new breakthroughs are just waiting around the corner, now that the path forward is so much clearer?